Good afternoon, everyone. Thank you for joining us in a webinar subject to macro-encapsulation and research kit. My name is Dr. Ophir Menashe. I'm a microbiologist of water and wastewater, also the inventor of the SBP technology. Joined to me today, Professor Adas Maman, which is the head of the environmental studies program at Tel Aviv University. And let me share my screen. Just a moment. I will upload the presentation. So today we will talk about new method in macro-encapsulation. And it's all about this. Can you see it? This capsule, SBP capsule. Okay. The SBP capsule is providing a new tool for studying encapsulation of microorganisms. It's actually a bioreactor that we are putting inside a bacteria or fungi culture in a suspended form. The capsule itself having a microfiltration membrane that having a pore size of 0.2 micrometers, which means that it's allowing to traffic only the organic matter, small organic matter or ions, but living inside the bacteria and not allowing other microorganisms to penetrate inside. It is a big capsule, consider a big capsule. It's 2.5 centimeter long and about 0.9 centimeter in diameter. And therefore, it's considered a macro-encapsulation procedure. This is a simulation of the capsule along with the culture itself. We can see the biomass in a confined environment, the confined environment made by the microfiltration membrane, separating from the host media. We can see microorganisms within the host media that cannot traffic across the membrane. For example, the free-swimming ciliata, which is much more bigger than bacteria, and the dissolved organic matter or ions or other nutrients can freely diffuse through the membrane according to the law of diffusion. This capsule the lifespan of this capsule is about up to 90 days, which consider very long. So think about it, how long you can do your procedures. It is also can be integrated inside a bioreactor, tube, Erlenmeyer, or every other tools that you will choose to operate. This is a slide that presenting the growth of bacteria culture inside the SBP capsule. The name of the bacteria is Rhedococcus zophri. It's a filamentous bacteria. You can see after 7 days, we can see starting of growing of the bacteria itself. And after 14 days, we can see that the bacteria is prosperate and achieving high density and mature. So we can see here accumulated of the bacteria inside in a filamentous and colony state. The second stage is how to use this kit. This kit, as you can see it here, encasing 25 SBP capsules along with a vial containing a sealing polymer. There is 3 major steps that we are doing with in order to operate the SBP capsule. The first step is to inoculate the capsule. We doing it by syringe or using a syringe and a needle and syringe the suspension, the bacterial suspension or fungi suspension into the capsule, creating an hole inside. We are usually inject about between 1 to 1.5 milliliter of the suspension. We would try to avoid spillage of the suspension from the oil, otherwise it will contaminate the external surface area of the capsule. On stage 2, we need to close the hole that we made. We are doing it by using the polymer, the sealed polymer. We are using a pipette, about 5 microliter of sealing polymer. We put the polymer above the hole that created hole, and waiting about approximate 5 minutes for drying. And we see that the polymer turns from clear color into white color. We're doing it about 3 times in order to verify that the capsule is sealed. And then we put it inside an RMI or beaker with a medium to activate the capsule. We're doing it for 48 hours in orbital shaker. We need to do it in order to allow the water to penetrate inside the capsule and dissolve the scaffold of the gelatin, so the gelatin will go in out of the capsule. It's taking about 48 hours, and I'm personally using it in MSN and 2% of LB. And then the capsule will change the position into vertical. And this is one of the signs that we know that the capsule is active. We have a video to show you, to demonstrate you about how it's happened. Just a moment. Okay. Let me just taking out this one and going back to the presentation. Okay, everybody hear me? Okay. Can somebody give me a signal that it's okay? You see the presentation?
We can hear you.
Okay, great. Thank you. So as you saw, this is how we are using the kit and how to operate it. And I will want to share with you about our experience in several fields of research. We are using this technology in life sciences, especially in microbiology, growing cultures, different types of cultures under different environmental terms. We also use it in biotechnology engineering, especially in fermentation, converting batch reactor into a continuous flow reactors for long-term fermentation, and achieving also a better product by separation of the production units, which is the bacteria or the yeast, and the product itself. It's saving lots of efforts, downstream clearance, and making our product better. Here you can see the bioreactor with the capsules, and we're putting inside a UV radiation. The capsule is protecting the culture from UV radiation. So what we achieved about it is to keep the medium, the host medium or the reaction medium, relative sterile, especially if we needed to do it in a long-term period for a few months. The procedure itself, as you can see, there is an inflow and outflow. That means that it's a continuous flow. This is one sample. The other one is the environmental engineering. We are doing in bioaugmentation, which means that we are using a special bacterial culture to target special contaminants. Professor Adas Maman will talk with you about integrating wastewater treatment approaches, especially physical, chemical, and biological together. And this is the time for Professor Adas Maman to present her experience in using the SVP capsules.
Thank you very much. So I'm very happy to be here first. I would like to first to introduce myself because my background is really important to what I want to say here. So first, I'm an engineer and I'm specializing in pollutant degradation using physical chemical processes, focusing on ozone, advanced oxidation processes. And actually, the capsules complement my work on the microbial aspects, especially in post-oxidation stages. And what I want to say is that the capsules really made microbiology accessible for me. As I said before, I'm a chemical engineer, so microbiology is not my expertise. And as an engineer, it was very convenient for me to use the capsules because it allowed me to simply, in a simple way, implement microbiology post-oxidation processes to achieve mineralization of the contaminants that I'm working on. And the results were consistent. And as I said before, it was simple to use. And again, I'm not a microbiologist, so for me, accessing microbiology was something that was very important for achieving degradation of pollutants in the aquatic environment. Using the capsules, we were able to integrate selective biomass into specific experiments for specific target pollutants. And we were able, for example, to measure the biomass level, to measure the degradation kinetics of the target contaminants. In addition to that, something that is very interesting that came out of this collaboration is together with, of course, Dr. Ophir Menashe, Professor Dror Avissar, and Professor Kurzbaum, we were able to actually develop a new IP from our collaboration. And that actually was using a combination of physical, chemical, and biological tools for microbial, for pollutant degradation. And this enabled also international collaboration beyond this specific collaboration. And I'd like to share another example. We also worked, for example, on degradation of contaminants in olive oil-based wastewater. We used, according to the recommendations of Dr. Menashe, specialized capsules that had microbes that could degrade polyphenols. So we were able to use to combine physical oxidation combined with microbial degradation to reach really good data and results in decomposing the polyphenols in the olive oil mill wastewater. This also led to additional research on degrading plastics. So there is a lot of potential and many options. And what I think that for me, and I would like to end with that, is that for me, microbiology was not approachable because I studied chemical engineering. I didn't work with bio-based degradation, only with chemical physical degradation. So using the capsules was simple, approachable, and I think, Ophir, that is my experience.
Okay, thank you, Professor Hadas Maman.
You're welcome.
And I really enjoy and collaborate with you in research aspects and finding new borders of this technology and in new aspects in research methodology.
Me too.
And now we will go to the section of QA. If you have any questions, we are open to answer it, of course. Okay, there is someone I want to ask. You need to raise his hand, and I know he have a question, he or she. You can also ask a question in a chat, write in a question in a chat. Okay, there is one question. Let me see it. Okay, does capsule have an expiration date? Okay, the capsule is provided in a dry state. It's unactivated. So the life shelf of the capsule, it's about 3 years each, something like that. Once it's attached to water, that's meaning you activate it and you wake the bacteria, the dormant bacteria inside. The lifespan of the capsule is between 2 months to 3 months. And this will define your experimental duration. We got another question. For a long time, it is possible to maintain operative the bacteria microorganism. Well, and the answer is the time that the lifespan of the capsule, I believe it's between 2 to 3 months. It's depending about the shear forces and the condition, the environmental condition that you put the capsule in the host medium. According to our experiments, in aggressive environmental conditions, it's about 2 months. In more relaxing flow rate, it's about 3 months. Another question, how much bacterial inoculum a capsule can sustain? I'm not sure that I understood this question. Once you are putting about 1 milliliter of bacteria suspension, it will sustain there. It will not go away. It stay there until the capsule is starting to breaking down. In the beginning, we will not see the breaking down of the capsule. It will be cracking. It will have a very defined cracking, but we will see the diffusion of the bacteria into the medium. So as long as the capsule is okay, we will not supposed to see any diffusion of bacteria into the host medium. The next question is, how do you use it in honey production? Well, I didn't did it. The people that made it is from the Technion. It was an IGM project. They took, I think, a bacillus, transplanted inside, or making a gene manipulation of the entire cassette of making an honey. We put the bacteria inside the capsule and host it in the right medium. Enzyme that's produced from this bacteria diffuse from the membrane to the host medium, and this is how we produce the honey without any bees. More questions? Okay, let me see if there is more questions. Yes, there is. Can the capsule be reused after the first activation? Well, this is an interesting question. I think it is. They can be reused. The thing is not to take it down out of the water or the medium more than 10 seconds. So if you can transfer the capsule, the activated capsule from one medium to another one very quickly, so you can reuse it. Other option, you can put it in the fridge and start all over, as long as you keep the lifespan of the capsule. So for your question, yes, Abraham, yes, we can reuse the capsule. Another question, is there a difference between yield, composition of secreted microbial compounds in a normal fermentation compared to a capsule? This is a very interesting, it was asking by Noah Ziklo. This is very interesting question. We don't have much experience with fermentation, and I hope for the answers for this question, you will provide it, Noah. More question, let me see. Yes, there is another by Adil Nasir. My question is the amount of the bacteria. I asked this question with reference to quorum sensing, as the bacteria is such confined environment can induce biofilm formation, and is this happens how and after how long will bacteria will return to planktonic phase? Okay, there is several questions here. First of all, about quorum sensing and bacteria concentration inside the capsule. According to our experience with E. coli, for instance, we're achieving a high density, more than log 7, in order to allow in quorum sensing. Also, we put bacteria with a luciferase system inside, a plasmid inside, and try to activate the luciferase reporting system by putting the autoinducer in the host medium, and we found that in about 20 minutes it's lighted. The other question is about the biofilm formation. Well, it is an option that the bacteria inside the capsule will form some biofilm, as you saw in the picture that I presented in Rhedococcus sofri. We saw that it's made a biofilm, a flock particle. Actually, it's made a flock particle inside. Pseudomonas also made a biofilm inside the capsule. Not all the bacteria may know is doing that. For instance, in E. coli, that has an ability to make a biofilm. We didn't identify it in the capsule itself. Let me see if there is more question? Yes, there is two more. Are there any conditions, such as temperature and pH, under which the porosity of the capsule can be altered? Well, I think it's very extreme temperature and pH. For instance, if you are boil it, the capsule, it will expand it. But mainly we are recommending to use the capsule in between 4 Celsius degrees to 45 or 50 degrees Celsius degrees, and pH between 5 to 10, and that will maintain the specificity of the membrane, or maintain the membrane itself. Another question: is it possible inject on selected bacteria into the capsule? Of course, this is the whole idea. This is the main idea why we are marketing this research tool, is to provide you the option that you can use your own bacterial culture or fungi culture inside this technology. What you are doing, you are growing your bacterial culture into a density and a stage that you want to use it. Take a syringe and a needle, put it inside, inject it inside a capsule, and seal it again. And then you have the capsule with the bacteria inside, your own bacteria inside. More questions? Additional questions? I'm open to answer.
Can I ask a question?
Sure.
Although we work together, as I said, I'm not a microbiologist, but I'm always curious. Can you explain a bit? You told me in the past about enzymes that can be released from the capsule. So can you explain that again?
Yeah, sure. Some of bacteria culture can secrete an enzyme outside the cells. It's called extracellular enzyme. The enzyme itself, it's a protein, it's catalytic protein, which is very small, much more smaller than a bacteria, so it can traffic across the membrane. We try it. Just put an enzyme. What is the kinetic rate of the secretion of the capsule, of the enzyme by the capsule? So the capsule also can manage as a control release for enzyme, a device for enzyme, and of course hosting a bacteria or fungi that is able to secrete an enzyme, and those enzymes will eventually reach out into the host medium.
Okay.
Let me see if I got additional questions. Well, I think that will be all. So I would like to thank you all for participating in this webinar. I would like to thank especially to Professor Adas Maman that was joining us. Thank you very much, all of you. If you have a further question, you can reach us with the email, and our email is in the invitation of this webinar. Hope to hear from you soon. Thank you very much. Take care. Bye.